What is going on, wonderful people? It's Medicosis Perfectionalis, where medicine makes perfect sense. Welcome back to my immunology playlist. In previous videos, we talked about your plasma and your blood cells. Your plasma is made of water, electrolytes, and plasma proteins. Your blood cells include red blood cells, white blood cells, and platelets. Some of these white blood cells are the lymphocytes. Your lymphocytes come in two varieties, B-lymphocytes and T-lymphocytes. The B-lymphocytes can change into plasma cells, and the plasma cells can give us antibodies, also known as immunoglobulins, otherwise known as gamma globulins. Each one of your antibodies has two light chains and two heavy chains. What are the different types of antibodies? What are their functions? Let's find out. Click the like button, click the subscribe button, and let's get started. This video is part of my immunology playlist. It's also part of my physiology playlist. For a more comprehensive discussion, check out my video titled Humoral Immunity. As you know, your blood is made of plasma and cells. Today's video is talking about the lymphocytes, which are type of the white blood cells. We'll also talk about the antibodies or the immunoglobulins, which are gamma globulins, plasma proteins. Where do the lymphocytes come from? From lymphoblasts. Where do lymphoblasts come from? From lymphoid stem cells, which come from pluripotent hematopoietic stem cells in the marrow of your bone. All of your antibodies, whether you're talking about IgM, IgA, IgG, IgE, or IgD, are gamma globulins. The functions of white blood cells were discussed before. Let's review them very quickly. Neutrophils fight bacteria, make pus, acute inflammation. Lymphocytes fight viruses, fungi, cancer cells. Chronic inflammation, not acute, chronic. They make granuloma, adaptive immunity, type 4 hypersensitivity reactions. They are also the cells that reject organ transplant because they don't like foreigners. Monocytes in the blood are the macrophages in the tissue. Macrophages are phagocytic cells. They eat foreign invaders. They are also antigen-presenting cells. We'll talk about this today. And they can make a granuloma. Eosinophils, ew, disgusting stuff. Parasites, ew. Allergy, ew. Anaphyaxis, ew. Type 1 hypersensitivity, double ew. Basophils in the blood, mast cells in the tissue. They release histamine, allergy, and type 1 hypersensitivity, asthma, all kinds of stuff. Let's compare between innate and adaptive. Innate immunity is natural, meaning you were born with it, but it's very non-specific. Acquired is adaptive. You were not born with it. You have to acquire it by training. And this is specific. Like training with whom? Training with the gym trainer? Oh, shut up. Training with foreign invaders. Types of immunity, let's go. Innate immunity, you were born with it. Non-specific, less potent, present at birth. No memory. The first response is the same as the second response, third response, etc. There is no difference. They respond to foreigners in the same way, like the acid in the stomach. Acquired or adaptive immunity, you were not born with it. You have to learn it. It is specific. The lymphocytes that target the influenza virus are not the same as the lymphocytes that target the pox virus, etc., etc. More potent, develop after birth, memory. Oh, I remember seeing this virus before, making your second response stronger and faster than your first exposure. We're done with the natural immunity. Let's dig deeper into the acquired. Acquired is naturally acquired or artificially acquired. Naturally acquired, you are passive versus you're active. You're passive, you did nothing. You acquired preformed antibodies from your mother through the placenta or through breast milk. Either way, you didn't do anything. Active, naturally acquired is when you get actually infected. Now your body will make brand new antibodies. Let's talk about artificial, blame your doctor, or thank your doctor. Passive, well, you didn't do anything. Your doctor gave you preformed antibodies. How about active? Your doctor gave you vaccines. Here's the lymphocyte. In the beginning, when they were young, they were very naive. No experience whatsoever. Then they will get introduced to reality. They will have to fight invaders. Once they recognize these foreigners and they deal with them, they will mature. They will grow the hell up. they will wise up and they will make memory cells and they will remember these organisms. So the next time I get invaded by the same organism, the second response is going to be stronger and faster. And this is called memory. Does the innate immunity have memory? No. The sequence of events have to be memorized by you. Lymphoid stem cells become T-lymphocytes and B-lymphocytes. Let's talk about the B-lymphocytes. When they mature, they give you two things: memory B cells and plasma cells. Let's talk about the plasma cells. Plasma cells secrete antibodies, which are immunoglobulins, which are gamma globulins, including IgM, IgA, IgG, IgE, IgD. So here's the B lymphocyte. When it was young, it was naive. After it got introduced to the antigen, it grew up into mature B lymphocytes and memory B cells. The mature B lymphocytes can become plasma cell to secrete antibodies. Each antibody has two heavy chains and two light chains. When it comes to the heavy chain, you can choose one of these. When it comes to light chain, you can choose one of these. For instance, this picture could be an antibody known as IgG kappa. B lymphocytes, plasma cells, immunoglobulins, IgM, IgA, IgG, IgE, or IgD. IgG, the most abundant, like ants. It's all over your body. Very small, therefore can cross the placenta. It's important for opsonization. It makes the bacteria tasty so that you eat it better, faster, and stronger. IgG is responsible for the secondary response when you see the same bacteria for the second time. Who's responsible for the primary response then? IgM. It's big, it's large, it cannot cross the placenta, but it's very good at fixing and activating complement. Remember we talked about complement in the last video, and complement always ends with the Incredible Hulk, the Mac, which will attack all kinds of foreigners. Why do you call it complement? Because it complements the antibodies function. Next we have IgA. IgA is in the mucosa. It decided to leave the bloodstream and ascend up there into your mucous membrane, mouth, nose, eyes, ears, vaginal canal, anal canal, etc. IgD, we don't know the function. It's produced first. We'll tell you in next videos that when the B lymphocyte was naive, it only had two antibodies. MD, it's like a doctor. The naive B lymphocyte is like a doctor, only having an MD. But then, when it recognizes the antigen, when it matures, it can make all kinds of antibodies. It can have M, G, A, D, and E. Tell me more about them. IgG, the highest concentration, the most abundant. Secondary response, small. What do you mean by small? Monomer, just one piece. Crosses the placenta. Opsonization. It can activate complement, but not as good as IgM. Also, IgG has antibacterial functions, antitoxin functions, and antiviral properties. Example of IgG is the Rh. And we talked about this in a separate video in my hematology playlist. IgM, primary response, produced first. Remember MD? Yep, the naive B lymphocyte was a doctor. M and D, produced first. It's very large. How many pieces? 5 pieces. Pentamer. Oh, that's very large. I cannot cross the placenta, but I'm very good at complement activation. IgM, complement. IgA, mucous membrane. You find in tear, saliva, wax of the ear, vaginal secretions, breast milk, etc. IgD, produced first, unknown function, maybe antigen recognition. IgE, disgusting stuff. Ew. Allergy, parasites, anaphylaxis, mast cell secreting histamine. Ew. Attacks of bronchial asthma. If you want to learn more about the mast cell and the story of histamine, check out my asthma videos on my pulmonology playlist. The story of immunology. This slide and the next slide are the most important slides in the entire stinking lecture. It's one of my favorite illustrations ever. Here's the deal. You have been invaded by a foreigner, like a bacteria. Okay, your immune system doesn't need the entire bacteria. I just need a piece. What's the name of that piece? Antigen. Where did it come from? From the bacteria. It's a piece of the bacteria. Thank you. Let me introduce this to the immune cells to tell them that this is foreigner. Okay, who's gonna carry the piece? Antigen-presenting cell. Carry it on what? On something called the MHC. Think of it like a waiter bringing you food. Who's the waiter? Antigen-presenting cells. Okay. What's the tray? The tray is the MHC. And what is the food? The antigen of the bacteria. The customer is always right. Who's the customer? The lymphocyte. The lymphocyte will recognize the food or the antigen. And then the lymphocyte will do one of three things, or all of them. I can eat the food, I will remember the food, and I can become plasma cell to secrete antibodies only if this is a B lymphocyte. Nice. Let's take a step back. You said that this is the antigen-presenting cell. Like what? It could be a macrophage, a B lymphocyte, or Langerhans cell. The tray could be MHC class 1 or MHC class 2. This lymphocyte could be B lymphocyte or T lymphocyte. T lymphocyte prefer to eat by herself. B lymphocyte prefer to become plasma cell and then to secrete antibodies, and then the antibodies will destroy the bad food. Why do you want to destroy the food? Because it's foreigner. Why do you want to remember it? So that the second exposure to the same organism or the same toxin is faster and stronger. Why do you want to secrete antibodies? Well, it could be to fix the complement, to bring the Incredible Hulk, the Mac. It could be to defend my mucous membrane, which is the first line of defense, anatomically speaking, against invaders. It could be to opsonize the bacteria or to opsonize the bacteria toxin to make it tastier that I can eat it faster, etc., etc., and so on and so forth. This story is beautiful. This is the key to understanding the entire book of immunology. Speaking of the tray or the MHC, it comes in two varieties, MHC class 1 and MHC class 2. MHC stands for Major Histocompatibility Complex, and I have a video on my channel titled Major Histocompatibility Complex, which you can find in this immunology playlist. So the waiter, I mean the antigen-presenting cell, presented the food, I mean the antigen. The customer, I mean the lymphocyte. We have two types of customers, B lymphocytes and T lymphocytes. B lymphocytes secrete antibodies into the fluids of the body, such as the plasma, tears, saliva, mucus secretions, etc. Fluids are known as humors. The ancient Greeks had a theory about the four humors: water, blood, yellow bile, black bile. Anyways, humoral means fluids. Okay. And we have another type of lymphocyte that cannot secrete antibodies. They prefer face-to-face interaction. I will face that nasty antigen head-on. That's why you call it cellular immunity or cell-mediated immunity. You're talking about a virus? I'll destroy not just the virus, the virally infected cell. I'll destroy the virus and the cell that the virus has invaded. What's that? Fungus? I'll destroy the fungus. Face-to-face, baby. Cell-mediated immunity. What is this? Cancer? I'll try to kill it face-to-face. I am not secreting any antibodies into any fluids. This is not who I am. Why do you call these B lymphocytes? Because they grow in the bone marrow, and in birds they mature in the bursa, so fabricious. Why do you call these T lymphocytes? Because they mature in the thymus gland. Tell me more about your T lymphocytes. Well, we have many types. We have T helper and T cytotoxic. What's the job of T cytotoxic? The name has the answer. What does toxic mean? Well, toxic. What does cyto mean? Cell. Toxic to the cell. What kind of cell? The fungus cell, the cancer cell, the virus infected cell. Thank you. What's the job of the T helper? The T helper helps all kinds of cells. I will help the T cytotoxic. I will help another type of T lymphocyte known as the T suppressor or the T regulatory cell. I will even help the neighbors, the B lymphocytes. Help them to do what? To grow into plasma cells and secrete antibodies. Thank you so much, T helper. You are impressive indeed. What's the function of the T regulatory cell or T suppressor cells? This prevents the T cytotoxic from killing your own cells, because these T cytotoxics are crazy. They can destroy your pancreas, they can destroy your own thymus, they can destroy your nose cells, muscle cells, etc. Who tells them to stay away from your own cells T suppressor or T regulatory cells. Stay away from the self. Go attack the foreigners. What if I have some kind of problems in my T regulatory cells? Well, your T lymphocytes will attack your own cells in a phenomena known as the autoimmune disease, such as lupus, for example. Some definitions. What's an antigen? It's a foreign substance that stimulates your immune system to produce an immune response. Why did you call it antigen? Because it generates antibody. Do you remember fibrinogen? Why did you call it fibrinogen? Because it generates fibrin. Same freaking concept. See, medicine makes so much sense once you understand what the flip you're talking about. What's a hapten? A hapten is a baby antigen, small antigen, very small molecule. On its own, I'm not good enough. I cannot generate an immune response. But a hapten, which is an outsider, plus a protein, which is part of your body, will make an immune complex that can generate immune response. Let's talk about the antibody. Who secretes the antibody? The plasma cell. Where did it come from? From the B lymphocyte. Where did the B lymphocyte come from? From lymphoid stem cells in the bone marrow. Let's go. Antibody is here. 2 light chains and 2 heavy chains. And there is a beautiful hinge right here and disulfide bonds connecting the two. We have a variable part and a constant part. The constant part is the same, doesn't change that much. All the options, all the actions, all the distinctions are here in the variable portion, which is also known as Fab. Why Fab? Because this is the portion that binds the antibody to the antigen. It's the fragment for antigen binding. Here is the antigen. Where will the antigen bind? Here or here? Well, the antigen always binds here, and this is the variable part, or the FAB domain. How about my own cell, like my own B lymphocyte or my own macrophage? It's going to bind here to the constant region. Where does the antigen bind? Here, in the antigen binding site, variable portion. Epitope is still antigen, so here. Where does the cell bind, like my macrophage or like my B lymphocyte? Here, near the constant portion. What's the function of the complement? To complement the action of the antibody, and it's going to bind here. Bacteria invaded my body. It's a foreigner. Let's take a piece called antigen. Let me introduce this. Who's going to introduce me? The antigen-presenting cell. Like what? Like a macrophage, B lymphocyte, Langerhans cell. Let's say it's the macrophage. The macrophage recognizes the antigen, and we have an antibody here. When the antigen and the antibody meet each other, we call this the antigen-antibody complex, or agglutination, or clumping. Now the macrophage will eat the entire stupid thing. Go to hell, bacteria. How can you make this entire process stronger? Bring in the complement to complement the action of the antibody. Don't forget, the first exposure is okay. The second exposure is even stronger and faster, because we're talking about memory here. B lymphocyte versus T lymphocyte. Who makes them? The bone marrow. Where do they mature? B cells mature in the bone marrow, T cells mature in the thymus. After that, they go work in their police stations, lymph nodes, spleen, tonsils, mucosa-associated lymphatic tissue, etc. In the lymph node, B cells live in the follicles in the cortex, but in the spleen, B cells live in the follicles of the white pulp. These are peripheral follicles. How about T lymphocytes? The T is always in the P. So the B is in the follicle, the T is in the P. Lymph node, paracortex. In the spleen, PALS. As we have said before, the B lymphocytes live in the follicles of the cortex of the lymph node, whereas the T lymphocytes live in the paracortex. You can learn more about the lymphatic system and the anatomy of the lymph node by watching these 2 videos in my hematology playlist. You can also find them in my anatomy playlist. Humoral versus cell-mediated. Pause and review. We just said that some of your antibodies, such as IgM and IgG, are capable of what? Of fixing complement, which means activating complement, which means pulling the trigger of the complement cascade. IgM and or IgG antibodies can start the complement cascade by activating the classical pathway. If you want to learn about the differences among the classical complement pathway, the alternative complement pathway, and the lectin pathway, please refer to my video titled The Complement System, which you can find in this immunology playlist. It's the best discussion of the complement system ever. If you like this video, you will enjoy my renal physiology course, 10 videos, 10 cases plus notes at medicosisperfectionalis.com. You can download it today. I also have an anti-cancer pharmacology course and an antibiotics course. You can find all of my premium courses at medicosisperfectionalis.com. There are more than 750 premium videos available on this channel when you click the join button and choose the highest tier. Please subscribe, hit the bell, smash like, support my channel on Patreon, PayPal, or Venmo. Go to my website to download my courses, notes, and cases, or if you would like me to personally tutor you. Be safe, stay happy, study hard. This is Medicosis Perfectionalis, where medicine, chemistry, math, and physics make perfect sense.